He's a professor of biochemistry. He is the Fraser Professor of Biochemistry here at UT Austin, and he's extremely well known for his contributions to the discovery and development of aptamers, which are nucleic acid binding species selected from random sequence populations. And they're used all over the place in point of care diagnostics to carry therapeutics into cells and as probes of cellular physiology. Maybe slightly less well known is that Andy co-founded UT's Freshman Research Initiative, the FRI, and it's frequently cited as a model for retaining students in STEM. So we're really excited to have him here today to talk about how he's been using machine learning, and I'm really excited to hear what he's up to. So thank you for being here, Andy, and please take it away.
Thank you. And so I'm going to be giving a talk along with an extraordinary graduate student, Danny Diaz, and Danny is going to do the projection. So Danny, I would say go ahead and share screen. Great. And he's going to advance slides for me. I noted, oops, there we go. I noted that there were a number of my lab members here, past and present, indicative of the great interest on our side in what you do. Of course, machine learning is transforming so many different areas of science, technology, and just everyday life. And so we feel like, we're getting in on the gold rush a bit, and we'll learn just an extraordinary amount from you, but we'll show you our own nascent efforts. And again, I would call out that those nascent efforts are largely due to people like Danny, who'll talk about his own efforts, and earlier efforts in my lab from Raghav Shroff and from Austin Cole, who I note is on the call today. So Danny, next slide. So my portion of the talk is mainly going to deal with what proteins are and how we've so far used convolutional neural nets to engineer them. And then I'll give it over to Danny, who's going to give a much more technical talk on how he sees the future of convolutional neural nets and machine learning for protein data sets. Next slide. So for those of you who don't know, and I know many of you do at some level, proteins are strings of amino acids. And in general, there are 20 natural amino acids. And all proteins are made, for more or less, from these 20 amino acids in different strings, different combinations. And the combinations fold up into three-dimensional structures, and those three-dimensional structures have biophysical properties. Like your hair is one type of string, and your skin, keratin, is another type of string, and so on and so on and so on. But basically, we just reorder these 20 monomers in different strings, and we can create pretty much all the stuff of life. So it's pretty cool. And I think you can see why even now this is a big data problem, because there are, n to the 20th possibilities for any given protein that we care to make, which becomes a large number, an intractable number very fast. And we need to be able to parse through those very large spaces in some more legitimate way than just poking at it. Now, that said, you'll see some examples of us poking at it in just a minute. Next slide. And so here are some of the remarkable structures that proteins can form. They can basically fold themselves into almost any shape. There's a really fun figure on the internet that's sort of like the periodic table of protein structures that shows all the different related structures in that sort of classic format. But again, because of the sequence, they fold into structures, and here are some of the structures. Those structures in turn dictate physical and chemical and catalytic properties. Next slide. Here's one example that's made our lives heck for the last year or so. The fact that SARS folds into a particular structure, its spike, and interacts with our ACE2 receptor, lets a virus into our system that unfortunately has caused us all to mask up for over a year. But at root, this is due to the sequence of amino acids in this protein and the shape it folds into. And one of the problems that I'm very interested in into the future is, and that Danny and others are working on, is can we predict what shapes go with what other shapes? And so that's sort of a silly Monty Python-esque level description, right? Stacking things on top of other things. But really, because we know what the shapes of amino acids are, we know something about the shapes of the strings, we can, I believe, future predict evolution. We can say what may come over the horizon, whether we're most likely to get infected by a virus out of a bat or a virus out of a civet cat or what have you. And so a area for the future, based on the work that I'm going to tell you about now, is to be able to predict these interactions and know before a virus even exists on this earth to expect it. Next slide.
Andy, can I ask a stupid question here? You can hear me, right?
Absolutely.
So let's say that somebody gave you the three-dimensional structure. So I give you, X, Y, Z, and fully describe the shape in 3 dimensions of these proteins. Are there interactions afterwards, a mechanical conversation, or are there chemical or other forces at play that complicate? So I'm saying, if I just gave you the shapes, is it just a children's locking simulator that you're running, or is there chemical and other processes going on that are not part of this?
That's a most excellent question, and again goes into greater depth than I just did. I sort of overlooked that most excellent question. It's both. The steric shape of a protein helps dictate its function, but down at the level of atomic interactions, which are sort of shown there more to the right, we have individual types of chemical bonds: hydrophobic bonds, salt bridges, hydrogen bonds, and so forth. And I'm not sure if Danny will get to this or not, but one of Danny's great interests is to use machine learning to extract the chemically salient features of a protein using saliency methods. And I'll leave that for him, but it's a variety of things, but we don't think it's so many things that a machine can't learn to tell us about them.
Okay.
Next slide. So this is just to indicate that in addition to the structure I just showed you, there are many, many, many structures. This is sort of a little bestiary of different types of folds in nature and how some of those folds play out in a relationship graph. And again, it's just meant to show you that there's a wide variety of folds. Interestingly, it's probably not all possible folds. David Baker at the University of Washington in Seattle makes novel folds never before seen in nature, and they're perfectly good and fine. Evolution has a tendency to rely on what has come before and to elaborate it. And so what you're looking here at from natural folds is just what nature has found most useful for nature. Next slide. Interestingly, as we look through the natural folds, we keep finding more and more and more and more and more protein structures. That's shown there on the left. This is how our database of structures increases, and it's been on an exponential path for quite a while. But interestingly, in blue there is the number of unique folds, and that's been falling, or on the right is the number of unique folds, not just in blue, and that's been falling off. It's as though we finally exhausted what nature can give us, and we're looking at variations on a theme at this point. And that's sort of comforting in terms of trying to predict into the future, in that we may actually know all the different basic folds, not the atomic level detail, but the basic folds may be almost completely determined at this point. Next slide. Now, I'm going to segue into some bio stuff, and it's only to show you how hard bio stuff is. I don't expect you to understand it in any great depth. One of the ways we will engineer proteins is essentially like a genetic algorithm. That is, we'll mutate the protein and we'll select for the variants that work. And this is a particular selection for a DNA polymerase. That's the enzyme that replicates every cell in your body and between generations. And we're just trying to make a DNA polymerase that works better under conditions that we want it to work better for. And so we will run this genetic algorithm, we'll take the mutations, and we have ways to make the polymerase essentially self-select itself. Like a computer virus, it can auto-amplify itself if it finds the right environment. So I don't expect you to understand in any great detail, but I wanted to show you, next slide, that the type of data we get will be to start with a protein shown there in the lower left. And then as we go through rounds of mutagenesis and selection, it will begin to collect mutations over its surface and eventually acquire a different function. And the function we've asked this protein to acquire is to be a so-called reverse transcriptase. That means it can replicate both DNA and RNA. Next slide. And interestingly, we've been able now to move this protein from working with DNA, and that's the chemical structure shown on the lower left, to working with RNA, that's the chemical structure shown in the middle, to working with 2'O methyl RNA. Now that's not really a natural nucleotide. 2'-methyl nucleic acids are something that people make. And so we've moved this enzyme between two different substances in nature to eventually work on this substance not in nature. And the reason I'm saying that is, next slide, it allows me to talk just a tiny bit about one of our other forays into computation, which again, if you press me on, I may not be able to fully expound upon, but fortunately I have people in my group who are quite good at this. This is an old Enigma machine, an old encoding machine. And of course, we talk about the genetic code, we talk about various ways of encoding biological information. But one of the things we began to think about is if you do encode information into biology, into DNA, how would you make it safe? Next slide. Well, one of the ways you make it safe is just to have a good encoding scheme. And so this is a so-called DNA fountain encoding scheme for taking binary information and encoding it into a short oligonucleotide that you could then store. And believe it or not, there actually is probably a decent industry in archival or glacial data storage in DNA. It's getting to the point where DNA is going to rival silicon, not for random access memory, but for being able to store data rather than in tape drives on DNA. Next slide. What we did was we took our specialty enzyme that we knew could read this specialty human-made nucleotide, the 2'-methyl, and we either encoded messages, very short messages obviously, in natural DNA, there on the left, or in the specialty DNA, there on the right. And what we were able to find was that once we'd encoded, only our specialty enzyme could read out the specialty DNA. A normal enzyme couldn't read the blue. The black could be read, the black and the blue could be read by either enzyme, but you needed our enzyme to read the blue. In other words, we'd not only done an informational encoding, we'd actually made a piece of hardware, for want of a better word, that was required to read that information out. And the person who did this in my lab, Jared Ellison, called this a cryptogenetic code. So that was just a slight excursion to show you A, how we engineer proteins without using machines, and B, some interesting things I think that we do in terms of using what we've made for data encryption. Next slide. But now back to the point. I will do a very, very, very brief and a very, very, very primitive introduction to how we've used convolutional neural nets, and then hopefully turn it over to Danny, who will do a much better job. My understanding of convolutional neural nets is they're able to extract features that are salient for a given type of thing you're looking at. So Danny and Raghav and Austin in the past have described this as, well, if you have a lot of pictures of cats and you know they're cats, then you can use convolutional neural networks, either 2D or 3D, to extract the salient features in a deep way and determine catitude. That's what I think of it as. It's the features of a cat, catitude. Well, we do the same thing, except we have these gigantic protein databases and the three-dimensional structures of proteins. So we're just determining proteinitude. And in doing this, we will eventually split this down, not just into, do you look like a protein overall, but what are your chemical and physical properties down at the sort of pixelated level. Next slide. And this is work that Danny has done and that he will be able to expound upon, but we take a protein structure and then we'll look at an individual amino acid, one of the 20 things in the code that folds up, and we'll extract that and the features around it, the chemical features around it. So we've eliminated all the blue, it's too far away, and we're just looking at that area of green, which can be a ball of different sizes. I think currently we're working at about 20 angstrom-sized balls around a given position. And then what we do is we say, okay, this is the ball that has a particular amino acid in. Remember, we only have 20. But now if we eliminate that amino acid from within the ball, we've essentially created an empty space, sort of a Taoist concept of a protein where you have this shape that determines what was there. And so now what we're doing is we're saying, can we classify that shape? And there only are 20 different classifications. You've either left out an alanine, or a cysteine, or an aspartate, or a glutamate. And so now we're looking not just at proteinitude, but alanine-itude, or cysteine-itude, or so forth and so forth and so forth. Next slide. And as Danny would have wanted me to say, and perhaps we'll correct later, that's a self-supervised learning task because we already know the answer. We know what should be there in the middle of that ball, and then we're retraining on that ball. But in addition, we're creating channels for retraining. And based upon work that we were basing this work upon, we're actually building on some other folks' initial neural networks. We use just the elements that would be present in that square or sphere. And proteins and biological materials in general have carbon, oxygen, hydrogen, nitrogen, and sulfur. And so we just look at the disposition of those in any of a variety of cubes that define our microenvironment. We also have included some other channels, partial charge and solvent accessibility, to again define channels that indicate the nature of one of 20 different types of microenvironments. Next slide. And because there are so many protein structures, 19,300 for this set, and we can sample somewhat randomly within those protein structures, we took about 100 microenvironments from any one protein structure, we can eventually build a training set of about 1.7 million such microenvironments, whose job it now is to discriminate amongst 20 things in those microenvironments. Next slide. Here's where, again, you all know way more than I do about how one uses filters in convolutional neural nets to identify features of a given set of objects. But to go back to what I know, we are now going to identify which of 20 amino acids fit in a pocket based upon making these filters from those 1.7 million different pockets and using a convolutional neural net to tell us the answer, to tell us not catitude, but amino aciditude. Next slide. And it works. I mean, no one was more stunned than me when it worked. The original network from Tong and Altman, where the paper we got this original from, was re-predictive of what should go in the pocket at about 40% rate. But by adding additional channels and by tweaking how we use those channels, we've been able to get that up to about 70%, actually more like 80% in some of the newer versions. So we are re-predicting across all of protein space 80% of the time what should have been in that pocket. Now, what's interesting is, what's that other 20% have to do with? Are we just not getting the right answer, or is there the possibility of other answers? And the reason we began to think that the net was actually not just telling us about what should be there, but what could be there, is that confusion matrix on the right-hand side, where we actually know from the physical properties of amino acids which ones substitute for one another. And sort of the off-diagonal predictions match up pretty closely with what we know from the physical chemical properties of amino acids. So we began to suspect that that 20% that we weren't predicting wasn't wrong, but that there might be a better answer from the network. Next slide. And that's exactly what we initially tried to find out. This is just a matrix of the various probabilities for the prediction of an ensemble of 20 amino acids at a given position. And so if you go down the left-hand side, the y-axis, those are different positions. And you can see in row F there that sometimes it's predicting the same thing. So row F, position 3, glutamate before in red, glutamate after in green, and proline and cysteine and cysteine, and proline, alanine and alanine. So it's predicting many of the same things. That it says the most probable thing to put here is the thing that was here in the first place. But at some positions, for example, there at the top, originally there was a lysine there, and our network is predicting a methionine should be there, lyse to methionine. And so it was doing this, the network was doing it with such regularity that we just had to test it for ourselves. Was the network actually just making mistakes, or was it telling us something new? Next slide. And another way to see this is on that structure at a given position, it's either predicting the wild type amino acid at the greatest probability, it's predicting a different amino acid, but one that's sort of chemically similar, or shown there in the lower left in yellow, that's a situation where it's not predicting the wild type amino acids, but it's predicting one that we wouldn't have predicted based on our knowledge of evolution and physical chemistry. It's predicting a change from arg, lyse, his, those are charged amino acids, to glutamine, which is a non-charged amino acid. So that seems weird, and again, something we could directly test. Next slide. I should also point out that Danny and his compadres have made it much easier for human protein engineers to look at what a computer sees. And I must admit, I did not fully understand what computer vision was for a while, because it was like, well, no, if I can see it, why can't the computer see it? I can see it. But in fact, of course, the way a computer sees things may be very different than the way a human sees things. And so this, while it looks very much like a representation that I could look at, is how the computer is looking at a given structure, where blue means it's repredicting wild type and red means it's predicting a mutant. So it's a way to look overall. And again, perhaps unsurprisingly to me, many of the predictions it makes for things that could be changed are on the surface of the protein rather than buried deep in its core, because the core of a protein is like a big jigsaw puzzle. It fits together incredibly tightly, and it is difficult often to make changes in the core. Next slide. We also have new tools that we're adding to our set of filters, one having to do with molecular dynamics, which is insanely computationally intensive, but nonetheless gives us the ability to predict motion over a very short range, because proteins, while we treat them as often as static structures, are actually incredibly dynamic. Now, this is where I said earlier we did even better than 70%. When we include these short, very short simulations of motion, we can get our accuracy for repredicting the wild type up to near 80%.
Andy, can I interrupt you for a second? Could you go back to the previous slide?
Yes.
I mean, you said it was predicting some amino acid that you were surprised by.
Yes.
And then you said you were going to test it. You didn't tell us the results of the test. And then
that's coming up. That's coming up. Yeah,
okay. And then what does that correspond to? Some sort of new type of protein
many new types of proteins. We're pretty excited about it. So
and what's the point of that? I mean, you know, I mean, why are you interested in creating kind of random new proteins per se?
Right. Danny, next slide. I think we're almost to the data. Yay, there we go, back up. So this is a little protein whose job it is to be blue. And we predicted over the surface of this protein how to make it bluer. And what we did was we weren't completely confident in our predictions at the outset. And so we just predicted a position and then made all 19 changes at that position. And along the top row there in red are the fraction of positions that made it bluer. And so at position 13, at position 18, at position 28, at position 114, some fraction of amino acids of the 20, besides the original one, made it bluer. And this was our initial test because on the bottom there were just some places that were just random. We just randomly picked places and the protein said, hey, could any of the 19 amino acids make this bluer? The point here is that the program, the algorithm, the neural net, was doing a pretty good job at picking places to make this protein bluer. There were more opportunities for going from not as good as wild type, blue, to orange or yellow, but the orange being the computer picked one and the yellow being just the out of a hat ones. So this was our first indication with a negative control, with a random pick as a comparison that the network actually was seeing things that might lead to improvement. Next slide. But what's sort of cool is that it picked all these positions and then we checked to see which of those positions could be mutated. And then as we began to mutate them in reality, not in computers, but in reality, we could put them together in an additive way. And when you sum all the predicted mutations, down at the bottom there was the protein the way it started out on the left and the way it ended up on the right. So we made a much, much, much bluer protein. Now, you may rightly ask, why do we care about a blue protein? In fact, protein fluorescent labels, which is what this is, are very useful. And we've been in discussions with companies about licensing this much bluer protein, but which has been made purely by predictive methods. Next slide. Here's another example. This is an enzyme that we were interested in, and we'd make it in cells and it would just drop out of solution. It would precipitate. It wasn't very soluble. And so we made a number of tiny little changes shown there by the blue bars that made it slightly more soluble and then summed those at the end to make it much more soluble. Now, this is definitely more so a biotechnology problem than even the make it blue problem, because many people make proteins at scale and they definitely need those proteins to be made in a way where they don't fall out of solution. So we were pleased to see that making something more protein-like, having greater amino aciditude, would lead to not just greater blueness, but also greater stability and solubility.
How do you know that affecting its solubility doesn't affect its function?
Because we also do enzymatic assays, which aren't shown here. But this particular protein does a chemical transformation, and its ability to do that chemical transformation wasn't harmed.
Okay.
I have a question here.
Sure.
In both of these cases, there is a goal that you have in mind. In one case, you want to get to the blue protein, and the other case, you want solubility, but your model is not aware of these goals, right?
That's right, yes. And that was one of the craziest things from my point of view of all this, and again, Danny can later correct me, this network is currently just looking at proteinitude. Hey, do you look like a known three-dimensional structure in nature? That's the only question we're asking it, breaking it down to the individual amino acid level. But apparently becoming more like a perfect form, more like an angel dancing on the head of a pin, results in a wide variety of properties being improved. Blueness was improved, solubility was improved. And as I'll show you in a slide or so, catalytic function was improved. In all ways, we're perfecting the protein, but we're not going for any one perfection.
Right, but in some cases, these goals may be in contradiction, right? So imagine that you had a redness goal. So you can't achieve both blueness and redness. How does your model decide to be blue rather than red?
Right, that's where having human experimental scientists comes in handy. When I show you these, for example, on this slide, this small set of improvements that we gang together, I'm leaving out all the ones that didn't improve it or that caused it, actually, good question, Adam, to be catalytically inactive. We, at the level of human examination, can make further choices regarding what we want or don't want.
So can I ask, following up on this conversation, in an unsupervised way, let's say I am clustering, I see some objects and I'm clustering them fully unsupervised, and then I take the centers of the clusters. Perhaps the centers of the clusters are the canonical pure blue thing and the pure red thing, and the centers of the clusters have been discovered without me even knowing what color is, right? Because
True.
I'm doing this in a fully unsupervised way, right?
Yes, that's right.
So maybe that's, when we were clustering movies, for example, and we found the cluster of all action films, the center of the cluster is the canonical, and we were indeed observing that there are some center movies indeed closer to the center of the clusters have the cluster properties more pronounced.
Right.
And this can be done in an unsupervised way, yeah.
Right, so I think Danny will comment on this later. One of the things we're going to try and do is, in an equivalent sort of way, see if we feed different data streams to the network. Let's say we have a data stream that represents proteins that are stable at high temperatures. We've now jiggered the game so it's not just all proteins in nature, but proteins with a particular property. Can we predict that property better? That would be similar to, although that's a supervised sort of network, nonetheless, we'd get at the equivalent question to, can we see all action films? So I think we're getting at some of that, and some of that may be pieced apart without training, without supervision. But I think Danny will talk a little more about how both via supervised and unsupervised methods we can get at that. To get to Danny, I'm going to go to the next slide, which I think is, this is just showing that relative to other methods that are based more upon biophysical techniques, we are predicting, our predictions are there in red, we're predicting very different substitutions. We're seeing our proteins in a different way than other people see our proteins when they do predictions using biophysical models. And I find that very interesting because it means that we don't really understand proteins, and it means that this new way of understanding can be especially valuable compared to things that people have done for a long time. Next slide. All right, so I'm almost through with my section, but I wanted to get to this, Adam, because this is why this is important. Blueness, great. Weird enzyme that's not as soluble as it could be, great. But in fact, we have real problems in this world that could be solved with biotechnology. One of them is plastics. There's a large team of folks here at the University of Texas working on trying to degrade plastics, trying to create sustainable plastic cycles so we don't all choke on our own waste. Next slide. And the way we're trying to do this is we're trying to create and improve enzymes that can degrade the plastics. And we actually have, again, a very robust program here that didn't initially involve machine learning, where we would try and identify the enzymes, screen for variants, a la the sort of genetic algorithm-ish approaches I suggested earlier, and then take those enzymes and actually do plastic degradation. But our work was thrown into high gear through the use of machine learning. Next slide. what Danny and others were able to do was, for this enzyme, which is the polyethylene terphthalase, or as you might think of it, the plastic water bottle eating enzyme, we were able to predict positions that, again, we had to test. It wasn't purely, every time we predicted it, we got a hit. Our hit rate's probably 10 to 20%, but given what we can do as biologists, that's huge compared to, the needle in a haystack that normally we'd go with from just random changes. But we were able to get to the point where, over a variety of different real world packaging, we can completely dissolve it. This enzyme will just completely dissolve it into its component monomers. Again, it'll take several days, but nonetheless, this is probably the best enzyme in the world right now for eating plastics, and it was designed by machine learning. Next slide. And this is where I hand it over to Danny. So I've tried to give you some idea of at least how a biologist views convolutional neural networks and the utility of machine learning, giving you a few examples of why it is useful and important to us. But now I'll let Danny talk about, into the future, how we really hope to work with you and to take these sort of approaches to a new level. Danny?
Well, hold on. Before you hand it over to Danny, let me ask one quick question from the audience here, and that is the recent Google AlphaFold, I think it's DeepMind, breakthrough on protein structure prediction related to your group's research?
Right. So given that I don't exactly know how AlphaFold works, and I'm not sure anyone outside of Google knows exactly how AlphaFold works, it's different. AlphaFold is taking the linear string of letters and folding it up and showing it's doing a much better job than humans at that. I have not yet seen Google apply their algorithms to improving a protein, but I'm sure they could do it. That said, even when they do it, I think that what we're doing is valuable in part because we bring our own experience as chemists and biochemists to the table, and we work hard at going back and forth between the experiment and the computation. Now, of course, Google X could do that easily, but right now, I'd say we're probably one of the better integrated groups in the world that can do at least some machine learning tasks and combine them with biological and chemical experiments. Danny, do you think that's a fair statement?
Yeah. So Google, essentially, their AlphaFold stuff essentially just makes our data set bigger, because we need structures and they can produce structures, and then we focus downstream of the structure. But
Gotcha.
yeah. So that was a great background on the biology, Andy, and all the cool stuff we're doing in the lab. So before I start getting into the nitty gritty, my background is synthetic organic chemistry. I think about enzymes, so enzymes that catalyze reactions from a chemistry point of view. So that's my background. But for the last few years, I moved over to the computer, and I've been doing a lot of software stuff. So this is kind of what I've been thinking about, the projects that we've been working on, and what's going on now, and what we're doing in the immediate future. So now that we have this proof of concept self-supervised model, there's a lot of things that need to be explored, need to be characterized, need to be better understood. A lot of you guys asked the right questions on some of the stuff that the second half of the presentation is kind of addressing. So I'm going to talk about some of the top bullet points here. So like data engineering, right? How do we represent proteins to computer, and are we doing it the best that we can? I like to think about it as the way that we're currently digitizing these proteins is kind of like video games in the 90s. I'm trying to bring it to video games in the 2010s, right? Have more realistic microenvironments so the CNNs can learn more realistic properties. So therefore they'll transfer to the bench or to real proteins more often, because you only saw the data of stuff that was working. Trust me, as Andy hinted, not all the predictions work, right? And we don't know how to tie a prediction to a functional property yet. So that's kind of the area where we're diving into. How do we know that this prediction is going to make it more blue and not make it more red, right? Can we embed some of that knowledge in there so the model can kind of help us out? These are all the things that we're trying to address and get deeper into. So we're going to focus on data engineering, data set curation to do these things. Data set curation goes really well with transfer learning. We can curate smaller data sets that we understand the properties better, so we can transfer learn a more agnostic model and fine tune it. Different self-supervised learning tasks with different architectures are going to be explored. unsupervised learning, clustering, trying to understand what's in these clusters, and the hopefully one day generative models. But all of these things have one thing in common, and that is we need a solid data engineering infrastructure to explore in all these directions. We essentially, I like to think about it like we need a Lamborghini to really drive around the town and explore everything that's going on. So we have built this library called ApBio. We call it ApBio because it teaches machines biochemistry. It's just how high school teachers teach high schoolers biology. It's a C++ data engineering library, and it enables for rapid data engineering. So making these microenvironments shown here, how we digitize the protein, and allows for us to quickly explore different CNN architectures. So ApBio, what it does is it takes the raw XYZ coordinates of proteins, filters out the microenvironment like what you see here, and then digitizes the microenvironment and then gives it to a neural network. That's the job of ApBio. So in designing ApBio, we set up some goals and some requirements. So we wanted it to be very modular, so we can functionalize different microenvironments, test out different parameters, plug and play, essentially. These data sets, right? These microenvironments, these several channel microenvironments are massive. Whenever you think about them in the millions to tens of millions to hundreds of millions, these data sets range from, at a minimum, hundreds of gigabytes to tens of terabytes. We can probably even make them bigger if we wanted to. And that makes it very hard to be agile if you're constantly having to store data sets that big, because then you don't want to delete them if you have to regenerate them. And then you need like an infinite amount of storage eventually. It doesn't allow for you to be very exploratory. So we wanted to move away from actually storing the data sets and instead just storing compressed representations. So like metadata, all the information you need to regenerate the microenvironment on demand. Next, we also need to make it accessible to people who aren't super intense coders. So we have a high-level Python API to expose the C++ data engine and make it for rapid development and testing. But the one goal of the engine is it must produce the data faster than the net can learn on it, right? So the data generation of per batch has to be faster than the back propagation per batch. Therefore, the time it takes to train a model is simply the time it takes to train the model. It's not the time it takes to generate the data and train the model. Does that kind of make sense? Okay. So, right, just going a little deeper into, right, the computational challenge of doing this, right? We need to populate this discretized space of atoms, right? We need to plug the atoms into their voxels, a three-dimensional pixel, essentially. So whenever we plug in the atom, is it here? Is it not here? That gives us a sparse matrix. But atoms actually take up space, volume. So then we need to blur the atom into this nearby voxels. So not only do we have to insert the atom, but then we got to blur the atoms in nearby voxels. And you can see as we explore different ways of representing the digitized space, right? For example, making the box bigger or doubling the resolution. So making each voxel smaller, all of that scales in a cubic fashion because we're talking about volume. So to do this, we just, each microenvironment is independent of each other. So we just utilized a multiple producer, single consumer design pattern to build the data. And we just use multi-threading and SIMD. Here's the architecture of ApBio. So essentially here you can see the panels are just different CPU cores. So an orange one, a blue one, a gray one. Again, right, multi-threading. And the way that it works is it's an object-oriented software. So we have the big players here is just this example maker. His job is to read in the files and filter out the atoms, right? What atoms go in the microenvironment and which ones can we ignore? That gives us an atomic collection that is simply just a stood vector. And then from this vector, we're just able to, we build a box builder. His job is to generate the actual boxes, right? So how big is the box? What is the frame of reference for the box, right? How are we normalizing our X, Y, and Z's? So that's what the box builder does. And then we have channel extractors. Their job is okay, now that we have this vector of atoms, we got to turn these vector of atoms into several channels. So what data goes in what channels? And then we have inference extractors, which if we're doing self-supervised or maybe supervised one day, if we have real labels, that's the job, is to grab the label. And then all of those kind of meet up at the discretized space, which is the actual box that is getting inserted with data. And then from this discretized space, it's in a format, it's a data structure that is optimized for data insertion, but it's not ready for machine learning libraries to train on. And then we pass it to the TensorFlow converter. We can easily swap out a different converter, maybe for PyTorch or something else. And that converts the data structure into a 4D tensor ready for libraries like TensorFlow. And then so all of this is multi-threaded. So we just have a concurrent queue that sits on the RAM, and we just populate the queue, all the threads. And then the model, all it has to do is ask for the next batch, and it feeds from the front of the queue. So that's essentially how our software infrastructure works. So here, just kind of putting some meat behind how we do things, right? This is not what our data sets look like, right? We went from having to store terabytes of microenvironments of these 4D tensors to just storing files of JSON objects of the metadata. So right here, this is one microenvironment. So this is protein 1IA1. So that's the protein file we're going to open. The ApBio goes to chain B. So proteins have multiple strings of amino acids. This is string B. And then we're going to go to the 24th character on the string. And that is the center of the microenvironment. And here was the amino acid in the center, the label. That's all the engine needs to know to recapitulate the microenvironment. So you just, these are essentially what our data sets look like. You can see how we can turn data sets of millions of microenvironments into just megabytes of data for storage. So here is speed benchmarks of ApBio. So you can see in blue is the time for back propagation for training. So that's running it through the net and then back propagating through the weights. And then in orange is the time to actually get the batch to the net. And you can see that we're not really waiting on the engine. It's a lot faster than the net back and then the learning. And this is how it scales with multiple threads.
Daniel, can I ask a question here?
Yes.
So I see you have a very sophisticated pipeline here in feeding the data into TensorFlow. And I'm curious, if you were just doing something much simpler, just dumping all the data in a CSV file and then loading it and training TensorFlow in a batch. I guess your whole point is that you're doing it in a streaming sense as opposed to a batch. Is there what's the time scale that it takes for training this current model? I guess.
Yeah. So if we have a 20 angstrom cube, I think an epoch takes us about 6 hours.
Oh, and how many epochs do you run?
We run about 5 to 10, 4 to 10, just depends.
Okay. Is it single GPU?
Single GPU.
Okay.
And then on the 40 angstrom box, a single epoch takes us somewhere between 24 or so hours.
I see. And your point is that batch generation is basically zero now. It's basically.
Well, yeah, box generation doesn't impact the training. That's how long it takes the net to just purely get the data to the GPUs, right, run an inference, back propagate. That is strictly that time because we're doing it in parallel. All
I understand.
The box generation is on the CPUs. All the model training is on the GPUs.
Right. You're basically saying it's all your data generation is free. Like it doesn't cost anything extra because the bottleneck is in the training anyway. So you
because while we're training on GPUs, the GPUs are just sitting there idle.
That's right. Makes sense.
Yeah.
So if the bottleneck is the training, then why are you so concerned with the data generation? Like as Alex said, I mean.
No, we're not concerned with it anymore. It's a solved problem. The concern was beforehand, it would take us like 7 or 8 days, maybe 5 days, depending on the data set to generate the boxes. And then if we realized, oh, we made a mistake on the box, it was supposed to be 24, not 22, or now we got to wait 7 or 8 days again.
I didn't realize that it was taking you 8 days data generation. But so what the bottleneck is just the size of the model, the size of the network?
Yeah, it's just the training.
Right, but I mean the size. I mean, how large are these models? Like, how many parameters do they have?
I think our current model is, I think, close in the 6 million ballpark region.
I see.
But like, we're gonna get to all that. I'm gonna show you
okay,
like
okay.
what's all going on there. So here is kind of the high-level Python API to make our microenvironments. You can see we just like instantiate some channels, then we add them to the builder. We're making use of the builder design pattern here. And then, right, so here's how we instantiate the physical properties. You just add them to the builder. And this is the Python code that's kind of responsible for generating these digitized representations. And here's actually all the code, right? So here is how we build the pipeline, right? All in Python. And then here's how we train the model, right? So abstracted away all the details of working with these files, parsing these strings of amino acids and actually figuring out if the atom's in a box or not. All of that has been extracted away. And then here is the software is in beta version, right? So this is just proof that it's up and running and working. We just retrained Mu Compute, got back to about the same accuracy. So now we're going to talk about what we're doing with it in the short term. So first, we're going to explore and experiment with how we digitize proteins. And then we're going to look at how we can apply CNNs to new protein engineering goals. So similar models, but because of how we explore and how we change, how we digitize it, now new biological problems we can address. So here is just purely fundamental exploration and characterization, right? What does the impact of different channels have on different learning tasks? What are some new channels? What are their impacts? What are different learning tasks that we can do? What is the impact of the size of the box, right? A 20 angstrom, 30 angstrom, 40 angstroms. How can we augment a data set to make a model more robust? So Andy was hinting at that with the molecular dynamics. That is essentially just wiggling the atoms a little bit, which is essentially just an adversarial example from a computer vision point of view. So these are all things that we're going to look at with this engine now that we have it running. Here's what we're going to do immediately. So our digital representation is incomplete. For one, it only sees atoms that belong to the 20 amino acids. So we're going to add things that are important in biology, like water. Water is pretty important. We're going to add phosphorus, because phosphorus is the other element that's also relatively abundant in biological systems. It's found in all DNA, all RNA, a lot of vitamins. So if we want to work with DNA, RNA problems, vitamins, etc., we need to be able to see the atom. We're going to add other molecules that DNA interacts with, right? So DNA atoms, sugar atoms, ligands. So ligands are like drugs, for example, or substrates to enzymes. Vitamins, like I was mentioning, ions. So metals are very important in biology. So like sodium ions, potassium ions, iron cations. So here, for example, is a microenvironment or a protein where our net is just not useful right now. And this is an enzyme that has bound vitamin B6, shown in gray. So it's a vitamin. So to our current model, it won't even see it. It has a phosphorus atom here in orange, wouldn't see it. And it's coordinating a zinc ion right here in gray, also wouldn't see it. And all of these red dots are water molecules that are key to how all the other atoms are oriented. Our model doesn't see it. So these are all things that we will be able to see here in the near future that would really complete the picture and allow us to address new biological problems. The last thing that we're going to experiment with, right, is whenever they crystallize these proteins, they also kind of measure the wiggle at a per atom basis, and they put them into these files, and they're called beta factors. So we already have the data there, might as well add it into a physical channel and see how the average movements of different atoms as a function of where they are located in space helps predict what should be mutated, what should not be mutated. So here is one of the first applications once we address all of these holes in our data engineering. And that is protein-protein interfaces, which is what Andy was alluding to a few minutes ago. So here is, again, the SARS-CoV-2 receptor binding domain, binding with the human ACE2 receptor. Here's the interaction. So here are all the chemical and steric interactions that occur. But so if you can see right here in gray, and right here in gray, there's a lot of sugars at the interface. Our model currently doesn't see those sugars. So that's why like these things are very important for all of these different applications. We will unlock being able to predict things really well there. So the goal there, right, is to help, as Andy was saying, like predict evolution, predict what are new viruses that can be zoonotic, essentially jump from an animal to a human, and how can we quickly develop proteins that are the lock to the key, right? The opposites, right? The opposite shapes, so we can quickly deviate the virus from our cells, but towards these therapeutics or these antibodies to help protect human health. So one way we can do this, right, we can generate an agnostic model, just how we kind of trained already, but it has a complete picture, maybe expand the data set some, so it's more balanced when it comes to metals, sugars, DNA, et cetera, et cetera, et cetera. And then we can just curate a data set and transfer learn it. And this data set would be of protein-protein interfaces. Maybe if we have enough structural data, it could be strictly of protein interfaces of human proteins with viruses. And if there's not enough data for that, well, we can add in other things like just regular protein-protein interfaces. But these are all ideas that we can rapidly test by just generating these JSON files, right? Just add microenvironments, swap out the proteins, go back to the command line, start training instantly. So now those are like the short-term goals. By short-term, I mean by like end of the semester and summer, right? That's what we're trying to approach. This is more like my PhD thesis goals, what I'm trying to address before I graduate. And that is looking at protein latent spaces, right? Clustering microenvironments, clustering the chemistry that proteins display, and seeing how it's distributed in this lower dimensional space. So Andy, this is a slide from earlier that Andy showed, all the different folds, right? Where there's millions and millions of known protein sequences, but there's less than 1,500 folds. And I don't think we've discovered a new fold in quite some time, definitely like years. So here's the same question from two different perspectives, right? Something that Andy would be really interested in is, can we learn how nature organizes protein chemistry into a finite number of folds, right? Why do so many sequences fold into such a small amount of similar shapes? Well, from a computer science or machine learning perspective, what Andy is low-key asking is, can we learn the protein structure latent space? So here is the MNIST dataset latent space. Here's an autoencoder architecture. I'm sure y'all are familiar with this. And this is just showing how we have the bottleneck, right? This is what we really care about, is learning this latent space.
You should use Stella's autoencoder from 2 weeks ago.
Yeah, I would love to meet Stella and get her autoencoder. So this is kind of what we want to do with it. So utilizing it for anomaly detection and for interpolation. So here again is the MNIST data set of digits. You can see, because we know the handwritten digit, we have a label. We can kind of see when someone has sloppy handwriting and someone's 8 looks like a 3 because it's in the wrong cluster, right? Well, what is the equivalent of that in proteins? Whenever there should be an amino acid there and it's the wrong ones there, either because it was nature doesn't evolve proteins to be perfect, it evolves them to meet a fitness level, right? The proteins just have to get 70s on their exams. None of them get rewarded for being an A+ student. What we want are A+ proteins that can solve problems in the real world. So we can use these latent spaces to find all of these regions, all of these anomalies, and use them as essentially low-hanging fruit for protein mutagenesis. And then once we have a latent space, it's continuous. We could just interpolate in the latent space. So you can see as we walk in the latent space here, the chair, this is how the chair changes in the real world. So as we walk in the latent space, the protein will be going discretized mutations of amino acids. That's what we're trying to get to. So here is a hypothetical latent space. I literally just made Gaussian random noise and distributed on an XY plot, hypothetical data just to prove a point. We're going to have some clusters in the latent space. What do these clusters look like? What do they mean, right? What does each cluster represent? Do they have different chemistries? If I color each microenvironment by the amino acid closest to the center of the microenvironment, where do each amino acid land, right? For example, is this just a cluster of prolines, Or is this a cluster of where an alpha helix turns into a beta sheet, right? Like, how is it clustering? I don't know, right? And then why are some clusters interpolatable and some of them are not, right? You can see how we can kind of walk in between these two clusters, but this is clearly a divide here. If protein chemistry landed right here, it's essentially like landing in the middle of the ocean and it drowns. The protein won't fold. The protein has not been observed in nature. This chemistry has to be detrimental for nature. We don't know. These are all the kind of questions that we can ask and we can really find out. So here's another example. So here's a globular protein grabbed from one of the first slides. This is the protein that binds DNA, pretty small, probably like 220 amino acids. Here is a monster protein. This protein is probably 20 different strings of amino acids, all coming together, working together. This protein is responsible for producing all the energy in our body. And it's actually the reason why we breathe oxygen, because it's what drives this protein to a function. So this protein is embedded in a lipid membrane. You can see it right here, all of these helices. This is the membrane where this is embedded. And what happens is it kind of acts like a turbine. Protons flow through it. It spins to generate the energy to then make chemicals that you can use as energy sources. But point is, this is a transmembrane protein. This is a globular protein. They have different folds. Well, let's randomly sample microenvironments all throughout the protein and then map them onto the latent space. So if I grab 1,000 microenvironments from one protein, 1,000 microenvironments from the other protein, map them into the latent space, what I essentially have are protein fingerprints in their latent space. The unique distribution of their chemistry is their fingerprints. Well, now that I have two distributions in the same latent space, I can now essentially compare and contrast these distributions with a KO divergence, right? So here is the fingerprint of this globular protein. You can see it has a lot over here. Here is the fingerprint of this transmembrane protein. It has a lot of chemistry over here. Here's both of them overlapped, and I can physically measure their KO divergence. Well, now we can extend that idea even further, right? A lot of proteins have the same fold, but yet they have different properties. Some of them work in hot water, some of them work in cold water, some of them work in acid, some of them work in base, some of them work in strong salt concentration, some of them don't, but they'll have the same overall fold. How does that happen? Well, if we want to engineer a property, what we can do is, okay, I'm going to grab these 5 proteins. They all have the same overall fold, but they all work at like 70 degrees Celsius. They're all thermophilic. They're an extremophile. Well, I'm going to take those 5 fingerprints from those 5 proteins and get all the microenvironments that overlap. And that's what you're seeing here in red. This is an extremophile fingerprint. Now I can take my protein that I want to engineer. Maybe it's the protein that some pharmaceutical company needs working at a hotter temperature for their bioreactors. Well, we can, but it's mesophilic, meaning once you go above 40 C, it dies. It's not thermal stable. Well, we can now take their 2 fingerprints and do a KO divergence using the functional fingerprint from the thermophiles, and then take the mesophile protein, look at their KO divergence, take the gradient of the KO divergence with respect to each microenvironment, and we know exactly which microenvironments are the ones that we need to target on and focus. Essentially, what we know is these microenvironments here are actually unique to the mesophile, and these over here are unique to the thermophile. We know we need to replace this chemistry with that chemistry. And we can computationally interpolate those microenvironments, maybe in a stochastic fashion. I don't know yet exactly how to walk them over there, but you get the big picture. We have now a North Star on how to mutate proteins to incorporate a functional property. From only a handful of proteins, right? I can make an extremophile fingerprint by grabbing 3, 5, 10 proteins that have the property I want and just making a fingerprint of the property, as long as they all follow the same underlying fold. So here is a summary of my talk. Well, Andy and I's talk. Protein engineering can address a diverse set of real-world problems. We have shown that machine learning algorithms can accelerate protein engineering efforts. We built ApBio, a protein structure data engineering library written in C++ with a Python API. We are utilizing ApBio to extend self-supervised 3D CNN models to new protein engineering applications. and we're experimenting with new architectures like a convolutional autoencoder. And I know y'all are probably going to ask me, we do also want to look at transformers, but we're just not there yet. Some acknowledgements. So all this is really possible by our lead computational monster, right? James Loy. He's a beast, right? I can talk, the way we communicate is I can write C++ enough to tell him what we're trying to do. He looks at my code, says, I get it, thinks it's cute, deletes it, rewrites it in a much more robust fashion, really brings all this to life. And we just go back and forth together to make the chemistry and the biology correct, but also the software engineering correct. Rogov for building the original model. That's really inspired me to pursue a PhD in all this. And then Ross Steyer, a lead experimentalist to all the first proteins you saw. Hong Lu for working on the PETase, and then our funding agencies, Welch, NIH, and DITRA.
Okay, thanks a lot, Daniel and Andy. We're a little over time here, but that was a fascinating talk and very interesting discussion. Are there any other questions from the audience here?
We're happy to follow up with anybody by email.
Alex, did you want to ask something?
Yeah, I mean, if we're over, I mean, just
I'll give you time for one question.
Yeah, I was trying to understand the problem that you mentioned, which is very interesting to me too, on generators for we work with Stalgan too, you know, on human faces, but the problems that you described, if I understand correctly, you care more about disentanglement of the latent space, right? Was that like the central? So you want, let's say, the first coordinate of the latent vector to correspond to
thermal
something.
stability, for example.
What's
right?
That?
Like thermal stability, how stable is the protein in hot water?
Right, but these properties that you want to disentangle, you don't a priori know them, right? So it's not that you, because for example, in human faces, we have a latent Z that produces an image of a face. And we say that when I change Z1, I get the head to rotate. When I change Z2, I get the guy to smile or something. When I change Z3, I change the color of the hair. But I didn't a priori impose these things, and I didn't even know these things will appear. Right? They appeared in a purely unsupervised way. Now, one thing that we are doing that may be relevant to you and we can talk offline is if you know the property you want, let's say you want to change the color of the hair, if you can train a classifier that does this for you. So you have a separate problem. You give it a face and it tells you if the hair is you know, whatever two colors you want. Or you can have multi-class too, it doesn't matter. But you have a classifier that controls the attribute you want. Then in our work, we use that as a way to explore the latent space. So we basically stick the classifier after the generator, and then we say, oh, you want my face but blonde? No problem. We'll do gradient descent in Z to increase the logit of the classifier. So for example, we have been making people who look like frogs that way by guiding the latent space.
You use a different regularizer, right?
It's not a regularizer. I mean, it's related to a regularizer, but it's guiding the search in the latent space to move in the direction you want. It's not a simple direction. It's not a one sparse direction, which is what you would want, but you can transform it into a, so I guess we can talk offline, but my main point is if you know what you want and you can build a classifier for what you want, then we can talk about sticking that classifier after the generator to get these directions and maybe simplify them. So we can talk offline if that seems something.
I would love to talk about that. Yeah.
Okay. I don't know if I'm making sense. You just need to classify.
No, you're making sense. So we're way over time here, but